I'm Julian. I am a recently graduated PhD student at the lab of Joseph Freire at the Center for Cellular Immunotherapies at the University of Pennsylvania. I will be presenting our research that I worked on with Dr. Matthew Ho and Dr. Luca Peruso, where we looked at unique toxicities that are showing up in patients that receive BCMA CAR-T cell therapy for multiple myeloma. So the unique toxicities that are displayed at the patient level come in four categories. So there is rheumatology-related toxicities, there's pneumonitis, so lung-related toxicities. The ones that we studied the most in our project were enterocolitis, so gut-related, and by far the biggest category was neurological-related toxicities, and that showed up as facial palsy or Parkinsonism. So when we looked at the clinical level, what factors are associated with a higher risk for developing these toxicities, we saw that high expansion of the CAR-T cells in the patient are significantly associated with a higher risk of developing these toxicities, as well as receiving a specific CAR-T cell product, which is called cilta-cel, which is the second CAR-T cell product that has been approved for multiple myeloma. Most CAR-T cell products are bulk CAR-T cell products, so they're just CD3 positive CAR-T cells, and then whatever the patient has is CD4s and CD8 CAR-T cells, and it usually just is directly infused into the patient, whatever the levels are. There are some products that make sure to adjust that ratio of having it 1-to-1, but for our products that we were interested in, the BCMA CAR-T cells, for both ide-cel and cilta-cel, we just have the whatever the ratio is for the patients is back infused into the patients. And we found that patients that had a CD4 skewing are more at risk for developing these unique toxicities. Additionally, we also found that there was a predominant CD4 skewing in those patients, so patients that developed these toxicities. In our project, we've also are the first to define these toxicities as CAR-T cell immune-related adverse events, or CIRAE for short, so CIRAE. And we found later on in more experimental studies that a hyperproliferation of the CD4 CAR-T cells is related to cytotoxicity in the patients, and through biopsies from the gut in the patients, we see that the CD4 CAR-T cell infiltration into the gut overlaps with the antigen expression in the gut, thus showing that the CD4 CAR-T cell infiltration into the gut is what primarily is driving these toxicities in the patients, at least on the enterocolitis level. We also performed single-cell RNA sequencing of CAR-T cells that were isolated from the cerebral spinal fluid, so more related to the neurological toxicities, and found that these CAR-T cells are also predominantly CD4, and in the CSF had a unique population that was highly enriched in cytotoxic transcriptional programs. Currently, the way the project is heading is more towards a preventative approach. So what's been really exciting about this project is that it has direct translational application, in that our clinical team has used these findings to sort of stratify the patients of who are more at risk for developing these toxicities. So they look at the absolute lymphocyte count or ALC short in the patients, and if they have a really high level, then they stratify within that group also whether or not they have, they could stratify whether or not they have high CD4 CAR-T T-cell skewing, so that perhaps preventative preventive steroid application could help in prevention of these toxicities.
If our videos have helped you in any way, and you're able to, please consider making a donation to help us continue this important work.
Your gift will go 3 times as far when we reach $500,000 by the end of the year.
Every contribution, big or small, makes a difference, and we're deeply grateful for your support.